Right, thank you very much for the invitation for coming to present to you today. My name's Dr Cath Rees from the University of Nottingham, but also I am part of a company called PVD Biotech, and I'm going to be talking about the development of an assay which we use to detect mycobacteria. So the assay is called Actiphage, and we can use that to detect infections caused by these mycobacteria, such as Bovine Tb or Johne's disease. The test itself, Actiphage, as the name implies, is based on these bacterial viruses. So they're called bacteriophage or phage more commonly. And these were first shown to be bacteria eaters, because this image here shows you a picture of an agar plate where there's bacteria growing all over the surface. And where the virus has been added, they have broken open the cells and causes these zones of clearings, these plaques. So this is where this name bacteriophage comes from, bacteria eater. This is just some really cool imaging that's come out of some helium ion microscopy. So here we have bacterial cells, and these are little viruses binding to their surfaces. Now what these viruses do is to punch their way through the bacterial cell wall, and they inject their own DNA, take over the cell, and as part of that process, they make millions of copies of themselves and an enzyme that breaks open the cell to allow the new phage to be released. Now, this is the bit that's really important to us, because these bacteria, the mycobacteria, are very, very different to the sort of normal bacteria we think about, E. coli and salmonellas. Their cell wall is very different, and it is very, very difficult to break open. And because of that, it's difficult to detect them using PCR. They're also particularly difficult to culture in the lab. So even using our sort of traditional microbiology-based methods, it's very, very difficult to find them. So what we are doing is using this virus to infect the cells, break it open. That releases the DNA for us nice and efficiently. And then we can use that DNA to carry out standard molecular biology type detection methods, usually PCR based, that we can then identify which bacteria are present. Okay, so the first bacteria blood phage test that we developed used quite a complicated format of the assay. And this is something that was developed originally 20 years ago or more for detecting mycobacteria in human sputum looking for TB. But we adapted this for a blood assay. So we took the blood, and the first stage of the process is to purify the white blood cells, because in these mycobacterial infections, the bacteria live inside those white cells in the blood. So we then use the phage to detect the presence of mycobacteria. So this is the original format of our assay. And you can see it took about 18 hours in total. But that allowed us to count the number of bacteria present and then extract DNA from that to carry out identification and confirmation of which mycobacteria we had identified. Now, that's very complicated, and it's not what we now call Actiphage. Actiphage is a new improved version of that was developed in my lab along with Dr. Ben Swift, who's now at the Royal Vet College. And the advantage of this method is that we got improved sensitivity without loss of specificity. So you can see the first stage of the assay is all the same: take the blood, purify the white blood cells. We then carry out the new modification assay, which is all done in a liquid phase, no agar plates, no long incubations. We end up with purified DNA, which we can detect, again, using PCR-based methodology. There's nothing new about the PCR steps that we are using. They're the standard ones everyone uses. So this allowed us to do detection and identification within 8 hours. This new format made it more possible to do high throughput, do lots of samples. And because everything stays in one tube, it's much simpler. We would predict that it improved sensitivity. So this was our first paper back in 2013 when we published the method. And I'm going to talk first about Johne's disease, because this is where we've done all the original work with this assay. So we developed the method in the lab, and we wanted to show whether or not we could use this to detect Johne's disease in naturally infected animals. So we went to a herd with a known Johne's problem that was one of my collaborators in the vet school was working with, and so we had good access to farmers who were happy to take part in new test programs. And we also used a high status control herd where there had never been any cases of Johne's disease, because we wanted to see how, if there were sort of any issues with specificities, if we found mycobacteria everywhere, or if we only found it in the herd with Johne's disease. In the Johne's disease herd, we selected the cattle, we want to give ourselves a fighting chance here, that had 3 consecutive positive milk ELISA tests. And these, on the system they were using, they were given red status. So there was a blood sample taken for a MAP blood ELISA as a standard diagnostic. We also used some spare blood to use a commercial direct PCR test that was sold by a company called Tetracore, to see if we could detect any mycobacteria in blood using a commercial PCR test. And we also used the Phage blood PCR assay. So there's a sort of snapshot of the results. And the first thing I want to point out is that we didn't get a total agreement between the milk and blood ELISA results. You can see an animal here that actually gave us a negative blood ELISA result, despite the fact it had 3 positive milk ELISAs. Now, when we came to the Phage assay, great news for us. All of the red animals were positive. In this form of the assay, we were counting the number of cells we detected, and there was less than 35 cells in each of the samples. And all of our control samples gave negative results. So this was a good first indication that things were working well. Interestingly, all of the commercial qPCR results were also negative. But the quoted limited detection for that method is 100 cells. We had less than 35. We really didn't expect any of those to be positive, and they all proved to be negative. So that was encouraging. So we carried on and tried a third set of animals. Now, on this occasion, what we had were animals that had been stratified according to their ELISA score. So the red animals had the highest. Green, amber's obviously a negative test result, and amber were near the cutoff. So they're in that middle. We only had one of those on this set of animals. Again, we did blood ELISAs. And you can see that in this occasion, we got even less agreement between the milk ELISA and the blood ELISA assays. However, when we did the Phage PCR, all of our red animals were red. And also, the amber animal gave us a positive result, and the green animal, one of the green animals, also gave us a positive result. We also attempted culture for all of these blood samples, to do blood culture. They were all negative, but again, the limited detection for culture, so as I said, they're very difficult to grow in the lab, is about 100 cells. We were only detecting less than sort of 50 cells in a sample. It was very unlikely that these were going to be positive for culture based on those numbers. So we're getting this lack of agreement between test results, which is what is the worst nightmare now when anyone is asking us, well, how do you know your test results are true? How do you validate something when the tests you're comparing them with have their own limitations? And this is a problem we've had all the way through this study, trying to put together enough evidence to convince people that these are not false positive results, they're real results, and that the pattern of results is meaningful. So doing this as a culture difficult. So fortunately for us, at the time we were doing these studies, a study was being carried on at the Morden Research Institute, Karen Stevenson, where they were infecting calves with MAP. Now, of course, because these animals were going to be culled at the end of the experiment, they could take tissue samples and do culture. And on this occasion, all of the infected animals were found to be culture positive, apart from this one at the top, number 1. When we did the phage assay, we found that 13 of the 14 culture positive animals, so this one was negative like this, but of the others that were culture positive, 13 of them gave us a positive phage result. So that was very encouraging. We had confirmed infection, presence of the organism, and our results were matching. Again, only very low levels of cells were detected, only 15 cells per mil. Now, as part of this study, a blood ELISA was also taken. And in this case, all of the ELISA results were negative, despite the fact these animals were culture positive. So this is the pattern we're seeing before. We're not getting agreement through different types of tests you use. So at this point, I thought, right, let's just think about what's going on with this disease and the assay. And there's a lovely paper that came out in 2017 from Judy Stable's group at the USDA in Ames in America, from the Animal Health Research Group there. And what they've been trying to do is to look at try and understand why we know that some of these test results are variable and differ at different stages of infection. So this is the model they've sort of drawn up in the paper, and it gives you a lovely visual. So this is sort of the gut epithelial layer. So this is the lumen of the gut with all the bacteria. And obviously, they're going to be excreted from the cow, yes, by natural processes, I suppose. Now, in the case there is no infection, we would expect to see no shedding of bacteria, no bacteria detected. In subclinical animals, we know that shedding is intermittent. Sometimes you will get positive results for feces, sometimes culture, sometimes negative. So what's the cause of that? So what they're saying is, in this model, we've got low numbers of bacteria accumulating in the tissue. Some of that is leaking into the gut, but the concentration on the levels of the bacteria in that mixture is very low, and we've got to take a sample of this. So as the sample is excreted, we will take discrete samples. Sometimes they'll be positive, sometimes they will be negative. And we see this sort of intermittent pattern of shedding. Once the animals progress to clinical disease, though, the numbers in the tissue, we know, get higher and higher. So the number in the lumen get higher and higher. And so it's more likely we will get a positive sample when we take a fecal sample for culture. So we would get this continuous shedding that increases with positivity over time as the numbers go up and up. So this explains our patterns of intermittent and continuous shedding. And the problem we have, though, is that there is a gap between no infection and subclinical. So in the very early stages, where there are very few bacteria in the tissue, very few in the gut, it is very unlikely we're going to get a positive result if we do culture, and so the disease is undetectable. And this is the limitation on this sort of culture method, where you need a lot of cells, evenly distributed in the feces, to ensure you're going to get a positive result. So sampling is difficult. But Johne's disease itself is also very complicated. What we know, that if you take experimental models of calf-to-calf transmission, that if you get animals exposed to MAP, you get high levels of those will show signs of infections within a short period of time. On farms with endemic disease, we know calves are routinely exposed to MAP. They've done studies of how much bacteria are in the environment on the farm. It's high. And so we know that they're there, and yet level of infections of individual animals is only 5 to about 20%. So it doesn't quite add up with what we see in these experimental models. So the term resilient has been introduced to describe susceptible animals that can be exposed to MAP, which then go on to show no signs of infection. Now, maybe they've got good mucosal barrier function, so the infection is established. Maybe they can clear it efficiently. Maybe there's other factors. It's not entirely clear, but it is clear that some animals are more resilient. And it's also clear that you get two forms of disease development: fast and progressive, quickly going to clinical signs, and slow and persistent, a maintenance state where you don't really get very fast appearance of anything clinical. So what's going on with the immune response during all of this? What we see is, if this is the point of infection and time going along here, that the first thing that happens is you get a cellular immune response. Okay, so this is the sort of thing we pick it up by a skin test or by a gamma test. So that goes up, plateaus, and then declines. And declines, interestingly, as clinical symptoms start to appear. Almost as if the animal has stopped controlling the disease and it now can't do anything more. It's got out of control. But at that point, the cellular immune response declines. Now, at some point during that process and after the cellular immune responses have sort of plateaued, we know that antibodies start to get produced. But you can see there's quite a time delay between infection and the antibody for the ELISA tests. Also, we need to get to a critical level before we can detect that response, and that's often only reached once we're getting close to when clinical symptoms are being detected. And of course, we know that the MAP shedding in the feces will slowly increase over time, as we've seen. And then again, it's in the later stages of clinical infection where we would get that guaranteed presence of MAP in the feces. So again, this paper that Julie Stable has put together to sort of review all of our knowledge about these tests, incorporating now this two patterns of disease, the fast progressing infections and the slow persistent infections. And it's quite interesting that if we look at the feces line here, you can see that these have been color coded on this scale. So anything red, the test isn't working. Anything green, it's working well. Anything orange or yellow is unreliable or an intermediate result. So in feces then, we can see that in the fast progressing disease, then fecal culture kicks in late to early to late, exactly as we've seen in the model. But in the early stages, it's not really going to work because of that very low levels in the feces. In slow persistent infections, it seems that fecal culture rarely gives a positive result. So fecal culture is the gold standard to ask the question whether or not your animal is infected. If it's a slow persistent infection, it's not going to work. Similarly, with the ELISA test, we saw this sort of progression towards the point where the disease, the test becomes useful. And again, it seems to be, you get that progression in the fast progressing cases of going from unreliable up towards intermediate or strong. Whereas in the slow animals, you get a bit of an unreliable response early, disappears in the middle, but comes back again late on. And again, interferon gamma doesn't seem to work terribly well for MAP. And this, remember how this is disease specific in either form of the disease. So what can we say about the reliability of phage tests? Is it any different? So this is some results we got for taking blood samples from animals, and we were counting the number of bacteria detected, and we had sample 1 and sample 2 from the same animal tested on the same day. And you can see we get a nice linear response here. So an R squared value in 0.9, it's pretty good. Shows us a good correlation between the numbers we get in the two samples. However, when you really look at it, okay, when we get at these low levels down here, the sorts of levels we would see early on in infection, the data gets a bit bouncy, as in sometimes it's positive, sometimes it's not going to be positive, okay? So reproducibility, like all the other tests, is going to become difficult at those very early stages of infection when levels are very low. However, what we know is when we get a positive signal, I have detected the DNA from the genome of the bacteria that causes the infection, and it is in the blood of the animal. It's not got there by fecal contamination. It's not passed through. It is circulating in the blood. And I know exactly what it is because we detected it by PCR. And so even though we may get intermittent results, false positives are unlikely. Okay, so just to conclude from that part of my talk, we know that phage PCR can be used to detect low levels of MAP in blood. When higher levels of bacteria are present, results are going to be both reproducible and specific. At very low levels, we do get this stochastic variation in number. So some samples may be positive, some samples may be negative when we're taking these very small blood samples, 2 mils. However, we know that Actiphage can detect infection before a detectable immune response develops. We've seen that in both the calf study and in the original ELISA studies, and we've also seen it in work we've done since. Johne's disease is complicated, so the take-home message is that different test results won't always agree, and depending on the stage and speed of disease development, that problem may be greater or lesser, okay? And this is always difficult then when we're getting to the point of trying to get our tests fully validated and understand exactly how our test compares with other methodologies. So for the last part of my talk, I'm just going to talk about bovine TB. This is the paper we published in 2016, which basically says we can use the same method that we use for MAP to detect bovine TB in cattle blood. The title says RPA. RPA is just another version of PCR. So if you see RPA, PCR, just use them interchangeably. So this paper was important because, first of all, against really the current dogma, it demonstrated that bacteremia was established early in TB infections, and also provided direct evidence that skin test positive NVL cattle, non-visible lesions, are really infected because we could detect bovine TB in the blood of those animals. So just to briefly summarize what's in that paper, we started off following animals to slaughter that were skin test positive. The blood sample was taken post-mortem to allow us to do these sorts of tests. Obviously, because it was post-mortem, the carcasses were classified as either visible lesions or non-visible lesions. In the visible lesion group, we only had 13 animals, but 11 of those gave us a positive phage blood result. Whereas in the non-visible lesion group, there were more animals, but we only got 16 out of a total of 28, so only a 57% positivity rate. What we saw when we counted the number of cells we were detecting in these two different groups, that the number of cells in the visible lesion samples was almost double, exactly double the average number we saw in these numbers. Now, given that we understand that test results will get less reliable as the numbers go down, this kind of explains this difference in the percent positivity between these two groups. So overall, 66% positive, and we had that higher positivity rate associated with higher microbial load. However, that study, first study we published, was all done using the original test with the plates and the agar and the overnight and counting. And this year, we now just published the results of the same study carried out using the new simple test, the Actiphage method. Now, we said that going to that new Actiphage method should make things more sensitive. And what we found was that 95% of all the animals, these skin test positive animals, had detectable M. bovis in the blood. So it's much more encouraging that this method now is working. So these results here, the ones I showed you on the previous slide, these are the ones using the Actiphage, where 100% of the VL, and I think it was one animal in the NVL group, gave us a negative test result. Now, encouraging again, in our negative test group, doesn't matter the sensitivity here, everything stays negative. So increasing this sensitivity did not decrease specificity. We're still getting this good discrimination between uninfected and infected animals. So we talked about MAP. and the fact that the immune response is complicated. And not surprisingly, in TB, the immune response is also complicated. So again, this is a diagram that Martin Bordemeyer from AFA has produced many years ago. It's been reproduced many, many times. But what we know is that this is a sort of pattern. Here's your point of infection, here's over time, and we're seeing the gamma interferon response growing and the tuberculin skin test growing. Both of these plateau and then they decline. Again, we get this gap between the cell-mediated responses and the production of antibody, and the antibody coming up later on. And over all of this time, we're expecting in tissues to see increasing bacterial load associated with increasing pathology. Now, interestingly, pathology starts after increasing bacterial load, because there will be that point where there are organisms present and yet no clinical signs. So we get this sort of progression of disease. So the question we wanted to ask was, could we use our Actiphage to detect bacteremia before the SICCT response was fully developed, before we got to the point where you're going to get a positive test result? And this has been quite well publicised. This project we were involved with, led by Dick Sibley from the farm down in Gatcombe in Devon. And he was working with a farm with a long-term bovine TB problem. So what he proposed was he was going to try enhanced testing of these SIRs, I'll explain what that means in a minute, to identify infected animals that had been missed by the SICC test, to see if he could get rid of that reservoir on the farm and clear out the TB. So SIR for us was defined as animals that get a reaction to the bovine tuberculin, but the lump was less than the avian, and therefore they were not classified as IRs. So there was a swelling, clear swelling, but because the avian lump was larger, they were not classified as reactors. Okay. In total, there were 154 animals tested. Actiphage tests were carried out after each of the subsequent SICCT tests. There were 8 sampling points in 2015 to 2018, and a total of 321 blood tests performed. So to summarise briefly, this was the first period from October 15 to July 16. The blue bars, these are our lumpy animals. Okay, they're not IRs, because we're not allowed to test those. So he's just given this reaction, but the reaction is lower than avian. And the orange bars show you how many of those animals then gave a positive blood test response with Actiphage. Now, this process went on, and there was other control measures happening. And over that time, we still were seeing positive results, but it seemed to be declining. And at this point, the farm got their first clear test for 4 years. But we knew that not all of the TB was eradicated. So Dick asked for permission to carry on testing, and then things got a little muddy, and testing was suspended for a while. But eventually, after negotiation, we were allowed to restart the program in June 2017. And again, we were still finding animals with a TB in their blood, even though they were giving a negative skin test result as on the severe interpretation. So on average, MBO was detecting 64% of this cohort of SIR cattle. And at this point, there was the second TB test, so we stopped again testing. So what do we know from this? This tells us that we could actually use this single intradermal test using the one tuberculin, use phage tests in parallel to ask the question, is that a specific response because the animal has Mycobacterium bovis circulating in the blood? So this gives us this sort of ability to use a more sensitive version of the skin test where you're trying to clear out disease. Again, how do we know if our tests are real? Okay, it's very difficult to validate. So in the first 2 sampling points, there were 20 animals that were included in this blue group on these 2 occasions. And just to briefly show you the data, of these 20 animals, 13 of them gave a positive Actiphage test result both times. And then there was another group of 7 that gave us either a negative then positive or positive and negative, this fluctuating result. But again, stochastically, low numbers, that doesn't worry us. A positive is a positive. Those animals are infected as far as we were concerned. Now, on this second set, we also carried out attempted blood culture. It's not easy to do, but we were successful. And we can see that of the blood culture, 9 out of the 20 animals gave us a positive result. And really interestingly, remembering that I need lots of cells to be able to get this culture to work, most of them, I think 8 out of 9, came from the set where we had a positive blood result twice. So we knew we had the highest level of bacteria. And 2 of the animals in this group eventually became reactors. So it's all showing us that these really are genuine results. So just to complete the sort of story of this study, after the negotiations and the resampling, DEFRA asked for gamma tests to be carried out because under the use of non-validated tests in the UK for private use testing, regulated by DEFRA, you need to have done a gamma test before you're allowed to use anything like Actiphage. So retrospectively, these rules had come in and they asked us to do a gamma test. And obviously, based on the phage test, the farm was really braced for a high number of positive gamma results. So 49 SIR animals were identified in December 2017. And of those, 31 gave us a positive phage test result. So we were pretty confident, patterns were going the same way, that we get a lot of these animals and we can show that those animals are infected. However, only 3 of those gave a positive gamma result. Now, interestingly, it was 3 that gave a positive result with all 3 tests we tried. Now, going back to this graph that we talked about, remember what we talked about, this, it rises up, the cellular immune response with the gamma, and then it declines. And this idea of these long-term persistent infections seems to be, if you remember from the test, that the gamma then it becomes ineffective. And the question that we're asking is, in these farms where we have these long-term persistent infections, are we actually missing animals because of this pattern that occurs in the immune responses? And maybe we can use this combination of the skin test with a single tuberculin plus phage then to give you confidence you know you've got infected animals to help clear out these infections. And not surprisingly, as we finished the program, when we had their clear TB test, lo and behold, 2 years later, down they go with, 18 months later, actually, they go down with TB again on that farm because we knew it was still there, even though we had to stop testing. So that's the end of what I want to talk to you about what we do know about our tests and what we don't know, which is mostly the latter. We know that the phage-based method detects bacteria rather than an immune response to infection. The presence of bacteria in the blood indicates that infection has been established. It's moved from a peripheral site to it's become systemic. Due to the complexity of the disease, our test results are not always going to agree with conventional tests. And really what we need to do now is do some longitudinal studies of infection to fully understand which of the tests, not just Actiphage, which of any of the tests are going to be used at different stages of infection and can help farmers eradicate these endemic diseases. Some other good points of our phage test, of course. Blood tests can be used to identify infection in animals at any age. So we've done calves, newborn naturally infected calves, experimentally infected animals. And at any time, you don't have to wait because we're not looking at the immune response. You don't have to wait between tests. The test could be used for screening young stock to identify infection at earlier stage, so you don't carry on rearing animals that you really don't want to introduce into the farms. It provides better, different information to use as part of an eradication program. But also, it's another test you can use to screen animals before movements. And we have got farms who are using this where they really don't want to introduce disease into new areas and are using our test as an extra test to try and screen animals beforehand. Final acknowledgements, of course, Ben Swift, I say, now a fellow at RVC and sort of building his own research team there. Tanya Perahinick, who runs our lab for us, Karen Stevenson at the Morden, Wade and Catherine, who've been students working on the project and generating data. Of course, all of our sponsors, and not to forget the cows who have given us their blood samples to make all of this happen. So thank you very much for listening, and I hope that was informative.